Understanding genetics is a little bit tricky task because genetics as a subject is full of definitions, fundamentals, laws, crosses, even the concepts from mathematics like probability, permutation, combinations, and all. But if you really want to understand the genetics in a logical way, then the most important concept of genetics is the phenotype. Because modern genetics, molecular genetics, and classical genetics, all these three branches of genetics are actually revolving around the concept of phenotype and how the phenotype is being regulated. In today's lecture, I am going to discuss what is phenotype. We are going to understand the modern definition of phenotype, how DNA sequences and proteins are regulating phenotype, and we will understand the Mendelian phenotypes again by utilizing the knowledge of DNA sequences, proteins, and biochemistry. This is the most fundamental lecture of genetics as a subject because once you understand the real definition of phenotype and its implications, then it is very easy to understand and very easy to apply remaining concepts of genetics. So let's start with the definition which we know till now. So what definition do we study generally for phenotype? So the statement says the term phenotype refers to observable physical properties of an organism, or you can say how an organism is looking like. What are the properties which you can see from outside? What are the physical properties? These include the organism's appearance, of course. The short definition of phenotype is also the external appearance of the organism is called as phenotype, and it also includes developmental and behavior characteristics. So generally, till now we are studying the definition of phenotype in a way that phenotype is nothing but how the organism is looking like and what are the characters which are observable or which we can see. But since we are in 2022 and we are having the knowledge of DNA sequence, we are having the tools to understand the amino acid sequence of proteins, we are having the tools to understand or dissect the biochemical pathways. So we need a definition by which we can combine classical genetics and modern genetics to understand genetic traits, to understand biochemical properties of genetic traits, molecular properties of genetic traits, and how they are being regulated. So we will discuss a new definition or an interesting definition which works most of the time for the phenotype by using the knowledge of DNA and its sequences, by using the knowledge of protein structure, by using the knowledge of biochemical pathways, and by utilizing the great central dogma. So let's start making a definition for phenotype. To make the definition, we need the help of central dogma, which says that the DNA after doing transcription is giving us mRNA, and with the help of process translation, the mRNA is giving us protein. So RNA polymerase is helping in the process of transcription to create mRNA, and the ribosome and other machinery are doing the process of translation to give us proteins. Now, if somebody asks what proteins are doing, what are the functions of protein? So I generally say that nearly all the functions are done by proteins. We are speaking, there is some protein behind it. We are running, there is some protein behind it. We are thinking, there are some proteins behind it. A biochemical pathway is occurring because there are enzymes which are proteins. So every function, nearly all the functions are done by proteins, and this is our baseline to understand the definition or to understand the logic and concept of phenotype, that nearly all the functions are done by proteins. Now let's utilize this information of central dogma and the fact that functions are done by proteins to frame the definition of phenotype. So we are having a particular protein which is doing a particular function, and because of that function we are getting some result, and this result we are calling as phenotype. So now our definition of phenotype is the result of a particular protein function. Whenever a protein will be functioning, whether it is catalyzing a reaction, whether it is making a new structure, whether it is giving a new character, the result of that function will be considered as what? Phenotype. Now we are having two basic mantras or two fundamentals of genetics. The first is all the functions are done by proteins, and second is phenotype is nothing but the result of a particular protein function. If we keep these statements in our mind, then it is very easy to understand all the laws, all the complex concepts of genetics, whether it is alleles, multiple alleles, gene interactions, then molecular regulations, all can be understood by one logic, that phenotype is the result of a protein function. Now I will prove this definition by taking one very simple example, that we are having a particular substrate or precursor which is called as GA20, gibberellic acid 20, which can give rise to gibberellic acid, which is a very important plant growth regulator or plant hormone. And this gibberellic acid ultimately gives the tallness of the stem, tallness to the stem in pea plant, which was taken by Mendel, right? So we are having a protein which is called as gibberellic acid 3 oxidase or GA3 oxidase, which is catalyzing the conversion of this precursor to the final product, that is gibberellic acid, and then final product is giving the tallness. So till now we are knowing the tallness as a phenotype, but we have found the protein. Which protein? GA3 oxidase. So we are having a protein which is doing a particular function. Which function? Conversion of precursor GA20 into gibberellic acid. And because of this function, we are getting a result, that is tallness, and that is our phenotype. So our definition is fitting, that phenotype is the result of a particular protein function. So from now onwards, whenever we are trying to understand the concept of genetics, our first task should be to find out the protein behind that genetic trait or protein behind that genetic concept, and to find out what kind of function is done by that particular protein. Now we have the complete network with us. We are having a result, then we have to find out because of which function that result is coming, and then we have to go for that particular function is done by which protein, and we complete the whole genetics. Every time we are going to do that. Multiple alleles, we will utilize the same thing. Which proteins are responsible? Incomplete dominance, which proteins are responsible? Gene interaction, which proteins are responsible? Mendelian phenotypes, which proteins are responsible? Because again, I am repeating, phenotype is nothing but result of a particular protein's function. Now, the definition again. Now we go to the next definition, and we try to understand what are alleles. Again, every time we are understanding a genetic concept, we'll bring protein into the picture, because we have to focus, we have to build the whole genetics around proteins and phenotypes. So proteins are coming from mRNA, and mRNA are being transcribed from genes. So my key here is the gene. If I take a diploid organism, that is a 2N organism. What do you mean by diploid organism? A diploid organism where each gene or each DNA sequence or each fragment of DNA is having two copies, right? I am having the gene which is coding the enzyme hexokinase. How many copies I am having for that particular gene? Two. Okay, that's why I am a diploid organism. Now these two copies or these two forms of DNA, these two forms of gene are called as what? Alleles, where the classical definition says alternative forms of a gene. So in a diploid organism, every gene which is coding for a protein is having two alleles. Means it is having two copies of that particular gene, and both the copies are able to give the proteins, or sometimes they are not giving the proteins, we will see further. But yes, in one organism we are having two alleles for a particular gene, right? And alternative forms of a gene means those two copies can have different characteristics, we will see. Now of course, a gene can have more than two forms or more than two copies, but of course they will not be present in same diploid organism. I can have two copies of hexokinase, two types of DNA sequences which are encoding for hexokinase. Another individual will come and he can have other type of copies for the same enzyme. Another individual come, and you can say that for a particular gene there can be multiple types of forms or more than two types of forms, so we call it as what? Multiple alleles or so. But in a single diploid organism, every gene is having two copies, every gene is having two forms in terms of DNA sequences, and we are calling them as alleles. Now if these two copies are identical, then we are calling them as homozygous alleles. And if these two copies are not identical, then we are calling them as heterozygous alleles. So again, utilizing gene and proteins, we have reached to the definition of alleles also. A gene is going to code for a protein. In a diploid organism, the gene is going to have two copies. These two forms of this particular gene are called as alleles. If both of them are identical, then we are calling them as homozygous alleles. If both of them are not identical, we are going to call them as heterozygous alleles. More than two alleles are present for a gene? The answer is yes, but not in the same organism. In different organisms or different members of the particular population, right? In that case is called as multiple alleles. But today's discussion, we will be mainly talking about a single diploid organism. So we will be considering that every gene is having two copies or two alleles. Now we are going to understand the details of alleles, where we already know that in diploid organisms two alleles are there. These are two copies of a gene. Then this gene is giving us mRNA. These two copies are giving us mRNA. From these mRNAs we are getting proteins. Then from proteins we are getting a function, and the result of that function is what? Phenotype. So we have added one more thing, that phenotype is the result of a particular protein function, and that protein is coming from a gene which is having two forms in a diploid organism, which we are calling as what? Alleles. Now there are few other terms like dominant, recessive and all. We dissect those terms also, by keeping the central character as protein. So our hero is what? Protein, right? So in your life, if you find a hero, then it is easy to understand all the things. So in genetics, our hero is going to be what? Protein. Now, imagine there are two copies of a gene, two alleles, and we are going to call them dominant allele if they are coding for a functional or a active protein. So if a particular DNA sequence is giving us the functional or active protein, which is ultimately giving us phenotype, then that particular copy or then that particular DNA sequence or then that particular dominant particular allele is called as what? Dominant allele. What do you mean by recessive allele? That particular DNA sequence, which is the form of same gene, is not giving us protein or giving us a non-functional protein or giving us a inactive protein. In an inactive protein, ultimately phenotype is not going to come. Then we are going to call that particular DNA sequence, that particular allele, as recessive allele. So the word dominance is being utilized here when we are having a functional protein with us, which can give us what? Phenotype. And word recessive is actually a condition where a particular DNA sequence is not able to give us a functional active protein, and ultimately phenotype will not come. So phenotype is the action, or phenotype is the result of a protein action, protein's function. This will come from a DNA sequence, then that DNA sequence will go into the category of dominant. And the phenotype is not coming because protein is not there, then that particular DNA sequence will go into the category of recessive. Now, one more interesting thing is, generally recessive is nothing. The absence of protein is recessiveness, means the absence of dominance is what? Recessiveness. So for us, dwarfness in pea plant is not a character, not a phenotype. The absence of tallness automatically becomes what? Tallness. So to make something tall, you need a protein. And if that protein is not present, then the same thing automatically becomes what? Dwarf. To make something dwarf, you don't need anything, right? So if that particular protein action is present, then we are having dominance. And if that particular protein action is not present, then we are having recessive condition, right? Now, we are again taking a diploid organism who is having a gene, and that gene is coding for protein. Now, this diploid organism can have three types of conditions. This gene is having two alleles. The first condition is both the copies of DNA sequence are giving us protein. They are identical, making functional protein, and ultimately giving us what? Phenotype. We just write them as capital A, and we call this condition as homozygous dominant. So what is homozygous dominant? That a person is having two copies of a gene or two alleles of a gene which are identical, or both of them are giving what? Protein, functional protein. And that functional protein is giving us the result, is giving us a phenotype. And if the protein is present and giving us phenotype, and that protein is coded by both the copies, then the condition is called as homozygous dominant. Or the person is having one copy which is able to give us a functional protein or the result, and the second copy is not doing that, but still the phenotype will come because one copy is still making the protein. We are going to call this condition as heterozygous. What is heterozygous? Two different copies or two different forms of the same allele or two different forms of the same gene. One is giving us protein and function, ultimately phenotype. the other one is not giving us protein and function, and no phenotype is going to come. Or the person can have, or the organism can have, both the copies which are not giving us a protein, or we are not getting phenotype, but the copies are identical. Then we just write it as small a, and we call it as what? Homozygous recessive. Homozygous means DNA sequences for that particular gene should be what? Identical. The word dominance will come if they are giving protein. Heterozygous means they are not identical. One is giving us protein, other is not giving us protein, right? And because of one copy we are getting the phenotype. The condition is heterozygous. Then both the copies are not giving us any functional protein, no result, no phenotype, but the copies are identical. We are calling it as what? Homozygous recessive. The word or is very important, because when in future lectures we are going to do the calculation related to genetic problems, then we have to use the concept of probability, and the concept of probability runs on two words, and or, right? So the same person cannot have all these three conditions simultaneously. The same person cannot have all these three conditions in the cells. The condition will be first one or second one or third. And I will be explaining in future lecture how to use this word or to do complex calculations of genetics. Now, questions. Till now, we are having two questions in our mind. The first question is, what are the examples of this definition by which we can understand the role of protein, role of DNA sequence in determining the phenotype, and how it is fitting to different examples of Mendelian genetics. The second question is, from where the recessive alleles are coming, how they are forming. So we will give the answer of these two questions by taking two very interesting examples of Mendelian genetics only. The first example is the Mendelian character of tallness. So I have already explained that the tallness is coming because of zebralic acid, and Mendel took the pea plant, and it is having three conditions. A plant can be capital T, tall plants, or capital T, again tall plants, small t, dwarf plants. So a plant can have any of these conditions out of three, right? All three cannot be together present in a single plant, right? So again, a probability based advantage will be there if we are understanding the plant can have this or this or this. Now, so I have already explained that the precursor GA20 is converting into zebralic acid by the action of the protein, which is an enzyme, that is zebralic acid 3 oxidase. This zebralic acid, once it is forming, then it is giving us the result in terms of what? Tallness. Now let's analyze capital T. What do you mean by capital T? It's a homozygous condition where both the alleles for this gene, which is encoding GA3, which is coding GA3 oxidase. So of course it's an enzyme, should be coming from mRNA. mRNA should be coming from genes. So that particular gene is having two copies. Capital T means both the copies are identical, and both of them are able to give the enzyme. The functional enzyme can come from both the copies. It means both the copies can make functional mRNA, the right mRNA, and that right mRNA is giving us the right protein. So both the copies are coding for a functional enzyme. The GA20 will convert into zebralic acid. Zebralic acid will give its result, and capital T will be a tall plant. Or the plant can have this situation, capital T means one copy is giving us coding a functional protein, another copy is not giving the functional protein. The condition is heterozygous, but still the protein is present. even if it is coming from one copy, right? So if the protein is present, this biochemical reaction will happen. And if the biochemical reaction is happening, GA20 will convert into gibberellic acid, which will ultimately give us tallness. So protein is present in the environment, protein is present inside the cell. Of course, the biochemical reaction will happen, plant will be what? Tall. That's why capital T small t is also a tall plant. But in the condition of small t, both the copies are identical, that is homozygous. But they are not able to produce the enzyme, the protein. If the protein is absent, what will happen? This precursor will not convert into gibberellic acid. And if the precursor is not converting into gibberellic acid, no gibberellic acid means the plant will not be tall. And if a plant is not tall, it will be automatically dwarf. That is the recessive character. So, this phenotype is the action or is the result of this protein's function. This protein can come from different type of combination of DNA sequences. If the DNA sequences, both of them are making the protein capital T, only one copy or one DNA sequence is coding for the protein capital T small t. Both of them are not making the protein, then no phenotype. So phenotype will come if the protein is present and phenotype will not come if the protein is absent. In some cases, in heterozygous capital T small t, if I compare this with capital T, then what will happen? If 100% protein is coming from this condition, then only 50% protein is coming in heterozygous condition. For some of the characters, 50% protein will not be enough. So there will be no true phenotype or complete phenotype. We call that situation as incomplete dominance, but we will see it in future lectures. But here, for the tallness, 50% protein is also enough. That's why the plants are still tall. And dwarf means the enzyme is absent, no gibberellic acid or basal production of gibberellic acid, no biochemical reaction, plant will automatically become dwarf. A very nice example of what? A very nice example of what? Our definition, which says phenotype is nothing but result of a particular protein action. Now, first question is solved. The second question is, what is happening at biochemical level? So again, the tallness is coming from gibberellic acid, which is coming from GA20, which is converting into gibberellic acid by the action of GA3 oxidase. So the dominant allele, which is able to produce a functional enzyme, is making GA3 oxidase, which is converting GA20 into gibberellic acid, and ultimately we are getting tallness. And if I compare that dominant allele with a recessive allele or with a DNA copy which is not able to make the functional GA3 oxidase, or it is making an inactive enzyme, what difference is there between these two proteins coded by the dominant allele or recessive allele? So if we look at the protein structure, then the protein structure of the functional enzyme is having alanine amino acid in the active site, which is replaced by threonine in the inactive enzyme. So the dominant allele is making an enzyme with active site with the alanine amino acid in the active site, which is functional and able to do this reaction. But the protein which is coming from recessive allele is having threonine instead of alanine. Because of that, the activity of this enzyme goes down drastically and it cannot catalyze this reaction. That is why tallness is not coming. So this is the biochemical explanation or the protein based explanation why the dominant allele is giving us tallness and why the recessive allele is giving us threonine. Now if I go back, one step back in the central dogma, I can see what are the changes at DNA level. So the DNA sequence of the dominant allele capital T is giving us a functional enzyme with alanine in the active site. The recessive allele, which is giving us an inactive enzyme which is having threonine instead of alanine in the active site. What is the difference between the sequences of these DNA fragments or these alleles? That on a particular position, the dominant allele or the dominant DNA sequence is having guanine, which is replaced by adenine in the recessive allele. So just by one nucleotide mutation, the dominant allele is converting into what? Recessive allele, and making an enzyme which is not functionally active. And if the enzyme is not functionally active, then the phenotype will not come, because phenotype is nothing but result of a particular protein action. Now we have solved the second question also. From where recessive alleles are coming? They are coming from dominant only, or they are coming from the DNA sequences only after some kind of mutation. Now we apply the whole thing again to another interesting examples of Mendelian characteristics, that is Mendelian character of seed shape. So the pea plant is having two types of seeds: round seeds, capital R, where both the alleles are identical, homozygous dominant. Then capital R, small r, heterozygous, still the seeds are what? Round. And small r, where the recessive condition is there and seeds are what? Wrinkled. So round, dominant, and wrinkled, recessive. Now what is our first task? First task is, this round and wrinkled phenotype should be the result of a particular protein action. People tried to find out that protein, and they finally found out that this particular protein is starch branching enzyme 1, SBE1. So if the starch branching enzyme 1, SBE1, the isoform of starch branching enzyme 1, is present, then the seeds will be round. So this enzyme will branch the starch and seeds will be filled, and they will look like as round seeds. If the enzyme is absent, then in the absence of starch branching enzyme, the seeds will be wrinkled, not enough deposition of starch, there is no filling of the seeds. So if I look at capital R, I will say this is a pea plant which is having two alleles, two copies of the DNA, which is coding for the enzyme SBE1, starch branching 1. They are identical, and both of them are making the functional enzyme, functional protein. We call this situation as homozygous dominant. Second, the capital R, small r, two copies of the same gene, two alleles of the same gene. One is able to make the enzyme, the second one is not making the enzyme. But still starch branching enzyme is present, it is coming from one copy, and that enzyme is enough to make enough branch starch, so the plant will be having still what? Round seeds, even if only one copy is working. Now small r, both the copies are identical, but none of them is able to make starch branching enzyme SBE1. No enzyme, no branch starch formation, no filling of the seed, and the seeds will be what? Wrinkled seeds. Right? Very easy to explain. You know, this concept of one copy is giving enough protein, another copy is not giving the protein, will be utilized till cancer biology, where we will see that oncogene mutations are enough in one copy, tumor suppressor gene mutations are required in both the copies. The same concept will be following till cancer biology, in immunology, in very complex concept of genetics, and we'll be utilizing one line again and again, that look at something and try to find out the protein responsible behind that. If you are understanding the function and structure of that particular protein, then other things are very easy. So we'll be following this line again and again, 100 times, 1000 times, infinite times in our brain, that phenotype or some character is nothing but result of a particular protein action. And here the protein is starch branching enzyme. If it is present, generated by both the copies, seeds are round. If it is generated by one copy, seeds are still round because enzyme is present, and that is enough to make a seed round. And if the enzyme is absent, none of the copies making the enzyme, seeds are what? Wrinkled. Now the second question. From where this small r allele is coming? Let's see. So the capital R, which is the normal wild type allele, which is giving us a functional starch branching enzyme 1, is giving us the phenotype, the result. But if I compare the small r, which is the recessive allele, is having an insertion mutation where in between the normal sequence of DNA, there is an insert of 0.8 kilobase pair, which is disrupting the sequence of gene, and there is no starch branching enzyme 1 production. So a normal DNA sequence, a wild type DNA sequence giving us functional enzyme, seeds will be round. A recessive allele is having a huge mutation in between. A DNA sequence inserted, it's a transposon like sequence inserted between the wild type sequence, that is 0.8 kilobase pair length, which is disturbing the structure of whole gene. The gene structure is disturbed, no mRNA will be coming, no starch branching enzyme will be produced. Recessive allele. If enzyme is absent, the function will not be there, and if the function will not be there, result will not be there. If the result will not be there, then there will be no phenotype, and no phenotype is what? Wrinkle seed. So there is nothing called as recessive or wrinkle seed. Absence of round seed automatically becomes wrinkle seed, or you can say absence of protein is automatically become what? Recessive. I am having one very interesting suggestion for reading. There is one very interesting paper, that is Mendel's genes toward a full molecular characterization by James B. Reed and J. Ross. You can go through this paper and you can find the information about other Mendelian characteristics also, how their genes were discovered, and these genes were coding for which enzymes, and you will learn other characters also with the new definition or a modified definition of phenotype, that phenotype is nothing but a result of a particular protein action. So I hope you have understood the logic of phenotype and how we should look at the phenotype from a new point of view, and how we should always look for a particular protein if we want to understand the concept of genetics, a complex concept of genetics. Before concluding the lecture, I am going to give you two scenarios where you have to apply your brain by utilizing what we have learned today. The first brainstorming situation is, in 1976, my grandfather was doing one experiment with rabbit skin color, where he was getting 75% rabbits with white skin color and 25% rabbits with yellow skin color. I am doing the same experiment in 2021, and I am not getting any yellow rabbit. I am getting all rabbits with what? 100% rabbits are having white skin color. The DNA sequences are same. There is no mutation. Just by applying the definition of phenotype, protein, absence, presence, you have to understand, you have to analyze why my experiment is giving these results and why my grandfather was getting yellow rabbit. And you have to find the answer, that what is the major difference between these two experiments. You can give the answer in our comment boxes, or you can write the answer to us. The second brainstorming situation is, a father is having homozygous alleles for a disease, and mother is also having homozygous alleles for the same disease, and the disease is not sexually connected. It means it is not present on X or Y chromosome. It is present on normal chromosome. So what kind of result we should expect? We should expect that all children should be having disease, because father is capital A, mother is also having capital A, capital A. So children should be having capital A, capital A. But surprisingly, all the children are normal. So you have to find out what is happening here, again by utilizing our knowledge of phenotype and proteins and presence and absence, and you can write your answers to us. These two problems, that particular reading, I hope you have understood the complete concept of phenotype, and now you will be having a new point of view towards genetics, which will make genetics as a subject very interesting, very exciting, and every time you will be reading a line about genetics, you will be looking for protein, which will make your life easier.